Good afternoon, everybody. My name is Rosalba Herrara, and my mentor is Dave. I am going to be a senior at Lincoln Park High School, and I work in the Dixon National Tall Grass Prairie Seed Bank. And my project is in Carex Viability. Right. So Carex is a Texan sedge and is native to sedge meadows. It has not been known if it has orthodox behavior, which means if it can be stored. Also, the male part is on the top and the female part is on the bottom. Alright, so currently million species of species have been stored in seed banks, and in the Chicago Botanic Garden seed bank, we store them at negative 20 degrees Celsius and with relative humidity of 15%. This experiment that I'll be doing will help the seed bank determine if actually storing the Carex stricta helps in the viability and future restoration of other endangered species. Right. So my question was, what effects does dry storage have on Carex stricta viability after harvest, and what X-rays affect seed germination, and will related species germinate similarly? My hypothesis is germination will not occur because research shows that seeds loses viability over time, and X-ray will not affect the seed because this machine I'll be using is especially for seed X-rays, and related species will germinate because they have similar growth habit and habitat. Alright, these are materials I used, the pictures of the column blower. So the methods, after collecting the fresh seeds, I put them under the microscope and use a forceps to pinch to see if they were full or empty. Then I put them in the column blower, which helps separate the empty seeds that are lighter from the full seeds. And afterwards I shed them to take the outer shell out of it, and I choose the best 25 seeds, and I x-rayed 25 seeds, and 5 of them were not full, so I excluded it out of the experiment. Then afterwards, I cleaned the seeds with 10% bleach solution to prevent fungi from growing in there. I sowed the seeds into Petri dishes with 1% agar solution, and I used a graph paper to evenly distribute the seeds. I put them in a growth chamber that was programmed to 30 degrees Celsius at daylight of 12 hours and 16 degrees Celsius at night for 12 hours. I recorded germination for 2 days per week. And afterwards, I tested seeds that did not germinate with tetrazolium chloride to see if they were viable. Okay, the picture on the left is the fresh seeds germinating pretty good, because you can see the sprouts coming out. And on the picture on the right is the frozen seeds that are also germinating equally in the same day, obviously. On the picture on the left is the seeds that grew a little bit too tall, so I had to take them out so they cannot be taking space. And on the picture on the right, it shows the X-ray image that I took. As you can see, some seeds are faded, which means they are empty, and some are white, which means they're full. So my day in Carex stricta, the frozen and the fresh seeds had similar germinations. I averaged them out and put them in a bar graph. As you can see, the fresh seeds are 24, out of 25, which is very good. And the frozen seeds were 23 out of 25, which is similar, like 1 seed off. And the fresh seeds that were 2 weeks were 22. So that means that they were in the mid-20s, very similar. Also, on the related species, they were quite ranging, since Carex aquatics and Carex lacustris were very in the tops, and Carex amorii and Carex lacustris were similar too. The aquatics was very good germination, even though it was dried for a very long period of 88 weeks. But the lacustris that was fresh collected did not germinate at all. So as you can see, the bar shows how if they would have germinated if we had more time. And we test that with we soak them overnight with trisolium chloride, and the next day I cut them open, and if they were red, that means they were viable. If they had nothing red, that means they were not viable. Okay, so my hypothesis was not supported since the frozen seeds show similar viability to the fresh seeds, and these results suggest that Carex stricta and related species exhibit orthodox seed storage behavior and can therefore tolerate cold storage preservation. Carex acostris did not germinate as well as I said before, but had positive results in the TC testing. The seeds that were exposed to X-rays germinate as well as the ones that were not exposed to X-rays, so that means it didn't affect them. And those are my works cited. And thank you all you guys who supported College Birds, Dave, Shelly, my instructors, Ben Murphy, and the Chicago Botanic Garden staff. Any questions? No? Okay. Well, thank you for your attention. Oh, yeah. Huh? How do it work? Oh, well, it's a solution, and you put the seeds in it for like about 24 hours. Every 24 hours, you cut the seed open and you inspect it under a microscope. And if you have like a little bit of red, that means that it did have cell respiration, which is viable. If it didn't show no red, then it means it was no viability. Any more? All right, thank you.